Good day. My name is Ryan Shepard, and I will be discussing the creation of a bioreactor system to produce human erythropoietin growth hormone. The research question proposed is there a way to produce purified erythropoietin growth hormone at a reduced cost locally? This question is in relation to patients in hospitals whom display low red blood cell counts. The normal procedure would be to administer erythropoietin growth hormone, which is also known as EPO. This growth hormone is produced primarily within the kidney, with only 10% being produced by the liver. This growth hormone is delivered in single-use cartridges, which have high cost and cannot be reused. So you may be wondering, how does this work? The erythropoietin growth hormone is responsible for being an important chemical in the process of erythropoiesis. This growth hormone causes selective proliferation of red blood cells from bone marrow stem cells. The amount of red blood cells produced by the body is directly related to the quantity and concentration of erythropoietin within the blood. Thus, if there is damage to the kidneys, there is a disease preventing, or if there is a disease preventing the production of erythropoietin, there will be a resultant decrease in the production of red blood cells. There has been an increase in the number of patients who display low blood cell counts. These patients have non-communicable diseases such as kidney failure, cancer, some anemias, and as well as HIV. Erythropoietin is produced in America and sold under the name epoetin alpha and darbepoetin alpha. The cost of such drugs, though, is exponential when considering importation taxes and the long waiting times of clearance, as well as delivery to the hospital. This would burden the state, which would use taxpayers' dollars to purchase such medicine, as well as it makes private care even more expensive and exclusive due to the high costs attributed. This study aims to provide a local source of erythropoietin by exploring the varying processes necessary for the creation of the drug. This local production, if viable, would allow for Trinidad to become a drug manufacturing center for varying growth hormones, as this process can be mimicked for all of the growth hormones within the human body. If successful, this project will allow for the upscaling of the product to industrial capabilities and will allow for jobs to be created. This study would also assess the necessary steps in producing a drug locally compared to the international acquired drugs of the same nature, and quality test the drug and ascertain the necessary clearances for distribution and international exploitation of the same drug. There are many related works that have studied such principles of creation of growth hormones within the body. An article written by Y. Beguin titled Erythropoietin and the Anemia of Cancer, which was published in the International Journal of Clinical and Laboratory Medicine, states the pathogenesis of the anemia of cancer involves combining shortened erythrocyte survival in circulation with the failure of bone marrow to increase red blood cell production in compensation. This article supports that there is inadequate serum level and response to anemia of erythropoietin. It has been proven that with the use of erythropoietin injections, as well as there is also a reduction in the need for transfusions, and it is also a safe and effective method in correcting anemia of cancer. Thus, in conclusion, this research has many different benefits which can be explored, and if there is a way to produce viable amounts of erythropoietin, it should definitely be pursued. This study has the ability to affect multiple stakeholders and has potential to be upskilled into thriving industry, and can contribute substantially to the economy via drug exploitation and job unemployment issues via personnel to work in the lab and at production facilities. Thank you for listening to my proposal.